All right. Hi, everyone. I'm Corey Watson. I have been co-chairing the GLDB working group with Andrew Collins for the last 5 or 6 years. So we're going to give you just a recap of what's been going on the last interval and tell you about our future plans. So for those who are newer to the community or just need a refresher, this is the mission we set at the AIRR-2 meeting many years ago for this working group. And the primary objectives here are really to promote the comprehensive and accurate identification, description, classification, annotation, curation, and consistent use of germline IG and TR genes and alleles across species, strains, and populations. A bit of a mouthful, but in short, basically try to make germline databases more complete and more useful and promote better analysis and better data sharing for the AIRR-C community. As many groups, we have an extensive email list, but the names shown here are those sort of reflecting the key individuals who have been contributing over the last interval regularly on calls and on specific objectives. Just a quick recap. So at the last meeting, we set goals for this interval. I'm going to basically go through these in turn and sort of give you an update on where we've gotten. In all cases, we've made progress in these. And again, I'll go through them as I do in turn. One of which I'm actually kicking over to the AIRR group, which you'll hear from shortly from Matt, which will save us a little time here. Okay, so the first one was the sort of very somewhat broad aim, which is really at the heart of what the GLDB is trying to do. So we really want to improve ways to both increase the efficiency and transparency and the effectiveness of how we curate and name genes. And this is definitely one of our big missions. This, however, is intertwined with existing bodies or groups that are already involved in nomenclature, principally the IUIS, IG, TRNH nomenclature subcommittee. So I'm going to kick the can down the road on this one because it's a bit of a complicated situation, but suffice it to say, we're trying to work with IUIS and basically improve this process overall. And I would need an entire 2 hours probably to go through everything that's happened in the last year. But suffice it to say, there's work underway here, and we're hopeful that that we can start doing a better job of increasing our efforts in this area. Second to that, one of the goals we set out last time stems from this recognition in the community that although historically we've really focused a lot on coding variation, there's a ton of variation outside of the coding regions across both IG and TCR loci. This includes both SNPs that are adjacent to gene segments, but also an intergenic space, as well as structural variation. And so the group has been working specifically with VDJ-base to start adding features to VDJ-base that allow us to step outside of the coding segments and begin trying to share and curate these non-coding variants. It's a bit of a busy slide, but to try to give you an idea what I'm talking about. And William Lees is your man if you want to learn more about this at the meeting, because he's done most of the development here. But the idea here is, we're thinking about our coding exons up here, which of course have alleles and different coding polymorphisms as shown with these blue ticks. But as we well know, there's lots of variation outside of these gene segments. And so William's been working really hard to try to develop features in VDJ-base for us to start cataloging these. And ultimately, the idea is that these will be able to link out to other databases, other genomic databases, and that this will provide a platform for us to both share and talk about these variants and what they're potentially doing in AIRR-seq data. Okay, then a big effort, and probably where we spent the bulk of our time and a lot of subgroup work in the past interval. These have been issues for us really since the beginning. But this last year, I feel like we really made a lot of headway. And that was to basically begin to compile together much more comprehensive sets of sequences for both mouse and rhesus macaque. Again, this stems from the recognition that we've had for a while that data available in IMGT, for example, was really incomplete with respect to the mouse strains represented, as well as the number of genes and alleles available for rhesus. And so we've been putting our heads together the last year to really compile as much data as we can that's available out there, and try to bring these together. And we're happy to note, at least for mouse now, that there are germline sets available on OgreDB for 18 different strains. And depending on the strain, there's data available for either IGH or IGHK and L. So that's pretty exciting. Again, if you need any more information on that, you can visit the link below, or you can find William. And then just very recently, through efforts with Chris Cottrell, Andrew Ward's groups at Scripps, and Gunilla Hedestum and Martin Corcoran's group who run ChemDB, we've started an integration of both AIRR-seq and genomic-based allele data sets, again, collated from over 70 animals. And so we've compiled all these together to try to make a broad collection of rhesus germline sequences that we're hoping to make available in OgreDB in the very near future. And the thing that is really important here is that, which I'll talk about in a little bit more detail in a minute, is that we've also began to use a new identifier or temp label mechanism so that we can actually name these sequences and allow for more consistent sharing of data between groups. Okay, so the other big thing that we've recognized is kind of forward-facing or future-facing, which is that the landscape in which we're operating under is changing in terms of the type of data that we're all using now to infer germlines or curate germlines. Specifically, the throughput and the genomic data types that we're using are increasing and becoming, in some ways, more complicated. What we recognize in a lot of this work that we were doing in GLDB was that current germline database schemas weren't really prepared or as adaptable as we might like them to be, to think about how we're going to curate sequences and link them to metadata moving forward, particularly as this pertains to using germlines from different data types. And so part of what we'll do, and I'll show you in a minute, is we've created a new germline database schema. And as part of this, as I just mentioned for rhesus, and this is true for mouse as well, this required us to also kind of create a new sequence identification or label tool or registry, which I'll go through. So the notion here was that historically, most germline alleles, say those that you would find on IMGT, historically came from either genomic reference assembly data or sort of targeted PCR-based Sanger sequencing and whatnot. Of course, the data landscapes changed rapidly in the last 20 years, and particularly in the last 10 to 5 years, where we've seen AIRR-seq and now lots more genomic sequencing coming onto the scene. There's a realization that there's all of this rich data and germlines available, and we don't yet have good systems to make the best use of those. And so that's holding us back. And partly that's holding us back because we don't have good data schemas to sort of link these data types together. And so one of the things that we developed here was a process by which we could bring in germlines.
Corey, you've got a 2-minute warning.
Oh, no, already. Okay, good. Well, that's good. I'm not too far off. And so we've developed a system now where we can basically use identifiers to help us link data together. And William can answer more in-depth questions as needed. And this is how it sits into the schema, which is relatively unimportant, but just to show you that we actually do have a real schema that's showing these boxes. And again, we could answer questions if there are any. Okay, and then we've also continued to work on questions regarding database versioning. So there's obviously always a push to make things more open, and everything that we're doing as part of the GLDB, particularly as this relates to database schema and releasing germline sets on OrderDB and DBJBase, these are all open license. So we're very happy to sort of try to be an example to the community there. So our goals for the next cycle continue to sort of solidify our work in mouse and macaque. This will include starting to integrate both genomic and AIRR-seq data sets. Continue to develop and try to push the use of the germline database schema in the community. Collaborate with DBJBase to keep on trying to build out features for cataloging and sharing non-coding variation. And then we're also interested in beginning to do a bit better job with community outreach, trying to bring in more stakeholders and partners so that we can bring in additional data from different species and try to get these into building better and more complete germline sets, and also get community members kind of implementing the germline sets and schema in their tools. And then, of course, a big area, which is true for most of us in the AIRR-C, is really thinking about sustainability and how we're going to maintain these databases through time. So we want to start looking for funding sources as part of this outreach. With that, Andrew and I are both keen to step down. So we've taken nominations through last Sunday. We have 3 individuals. So those who are in the Germline working group, you will see a poll for me later today. So we want to hold a vote over the next 2 days, which we hope to ratify 2 new co-chairs on Thursday as the meeting closes. And for anyone who's interested in joining the working group, please email me if it's in the next 2 days. Otherwise, feel free to reach out to Pam and she can link you up with the right people. With that, thanks for your time.
Any questions for Corey? Jamie.
You have to repeat it.
Hi there, really nice.
Oh,
Is this working? Okay, good.
Yeah.
Corey, really nice. I have 2 questions for you. One of them is with regard to your proposed leadership. Is the working group itself deciding on who their co-chairs ought to be? I mean, that's typically what they do. Or when you said you're sending out a poll, is that to all of the AIR community?
No, sorry, I wasn't clear. It's just to the GLDB working group.
Oh, great. Okay, good.
Yeah.
And the other thing I wanted to ask you about that's kind of come to mind over the past few days is as you're integrating these germline gene databases for, let's say, the mouse or the macaque, depending on where you get them from, there are different guidelines or, different rules that they use for putting something in. And as we know, in some databases, there's a lot of crap.
Mm-hmm.
So what I want to know is, AIRRC had the whole red light, green light thing going. Is there a way, or is it just too much work for people to say, okay, this is coming from this database and here are their standards. So we're going to assign this sequence, with this level of confidence. This one's coming from this semi-shitty database. And so we're going to assign this level of confidence. We can look at it a little bit further or whatever. But do you see what I'm saying? So that people get a confidence score when you do this integration or something like that?
Yeah. So I think a formal confidence score, we're not really anywhere close to that. But part of the objective of these germline sets is to really link each sequence, each germline to metadata. So that would include source information. So is it only seen in AIRR-seq? Is it only seen in genomic data? Particularly for like the macaque germline set we're getting together, there's going to be information about, the frequency which is observed, how many animals is it observed in, so on and so forth. So that's kind of the vision we have moving forward, that you'd have a lot of this kind of rich metadata associated with the germlines, and folks would have the opportunity themselves to take the germline set and kind of use that information to take out the sequences they want to use if they don't want to use the data set in its entirety.
I'm sorry, I'm being told we're way over time.
Okay. Bye.
Thanks very much,
Can I ask
Corey.
a quick question? Quick. All right, fine.
I'm sorry.
You can ask me online
already.
or ask William.
But I'm William. Do catch me or come to my poster or something. Very, very happy to talk further. Thank you very much.
Thank you.